Don't mess with my 1 minute, which is equivalent to 120 minutes of your prime time. Okay? This is simple philosophy. Yes? So in the end, I'll just write your attendance on the back of those who came late. Write your attendance on the back of this page. I'll put your attendance. Is it? Is that okay? Don't worry. Bismillahirrahmanirrahim. Introduction to physiology. We'll talk about, this is the primary book of physiology, which is the Medical Physiology Guidance and all. This is the textbook of physiology. There are other books of physiology, we call it the review book of physiology. There is a class. Denong is a review book. You have to read the basic textbook first in order to understand the review book. Because one page of review book is equal to one chapter of the basic textbook. So basic textbook is important because in basic textbooks they teach again and again the principles of physiology. And they relate the questions again and again. They relate the concepts again and again. They discuss the concepts again and again so that you can understand how the physiological principles are working in this situation. So most of the time there are lots of repetition, but you have to understand it, you have to learn that. So if you really, but you have to read the basic textbook first in order to understand the review book. So if you are directly jumping to the review book, you will not do justice with your understanding of physiology. It will be very much affected. The second thing is, I always, talk about so many examples, but I'll give you one example. If you do not, suppose you do not have a driving license, and if you want to drive a car, and if I show you 20 hours of lecture how to drive a car, if I give you 3 lectures how to drive a car and do not allow you to sit on the driving seat and drive the car, what is the likelihood of you passing the driving test? Very low. Because you have not done the actual test of driving, because when you go for the driving test, the instructor sitting next to you has a checklist with him or her, and he will check, you have done this, you have not done this, and so on and so forth, and based on to this assessment, you will be paid. Or likelihood of passing is very low. Similarly, you are given by lectures and practicals and then labs and other things. You are assessed by, remember my words, you are assessed by MCQs and SAQs. If you do not do MCQs and SAQs in your practice sessions, then the chances that as if you are going to the driving test without having a first-hand experience of driving onto the road. So please do the questions. These 2 books, the Guided Model Physiology Review has questions related to the topic. So go and read these questions after reading the book, after reading the lectures, okay? So please review this and then try to answer these questions. There is another BRS, Basic Review Series, is the book which is always there in anatomy, physiology, biochemistry, and so on and so forth. So the review questions are there. Please solve these questions and those questions. Once you solve these questions before coming to TBR. Solve these questions before, to make sure that you have already done these questions. If you have learned today's lecture, if you have learned read the book, and you solve questions, and if you solve all the questions correctly, it means you understand what is being taught into the lecture. So this is the best way to retrieve your, recall and analyze and memorize your information and consolidate your information. So that is very important. So learning physiology or any subject, if you do not solve MCQs, there are different levels of MCQs. Some of the MCQs we'll discuss while we do the introduction to TBL. I'll discuss. There is a pyramid. We'll discuss that over there. So please solve MCQs. And my assessment is, let me say this sentence here. If you come to the TBL and without solving MCQs, the chances that you will not get a good score. If you come to TBL after solving about more than 50, 60 MCQs, this is the safe line, safe number, from any resources, it means you are working hard and you will know the inside and out, and you can easily be able to understand and be able to solve those questions individually. So that is what my word of, I'm safe. If you really want to be safe on to T-bill, that is the best thing that do solve MCQs, do come to the T-bill before solving MCQs. Don't forget to solve the T-bill. There are two objectives. Am I very clear here? We'll discuss these objectives, and we'll also discuss what is physiology. Physiology is the scientific study of the normal body. It is scientific study. When we say it is not the study, it is the scientific study. Integrated normal function. When we say integrated normal function, we have the digestion, we have respiration, and this digestion and respiration, they are one way or they are different systems. But in a body, in a multicellular organism, they are integrated. So integrated function is the physiology. Scientific study of integrated function is the physiology. So function in isolation does not work. If you study digestion separately, okay, it's good, but you need to understand the digestion in the whole body, how it is being helping in maintaining the normal progression of life. So progression of life, you have to understand the whole bigger picture. Okay, so try to understand that. Okay. The other thing is, it includes physical and chemical factors. What are the physical, chemical, biochemical reactions taking place in the cells? Metabolism taking place in the cells. You need to understand. And that meant what are the substrates needed for metabolism? What are the enzymes needed for that metabolism? What are the end products produced by the metabolism? What are the consequences? What are the things which are extra produced? What are their effects? So all these things, they have their effects. You need to understand the bigger picture. So physiology is not understanding the smaller, minute detail, but if you have to relate it in contextual way from the clinical aspect, and that is what physiology is. Okay. So it explains the contribution of organ systems to the homeostasis. There is a term, we call it homeostasis. We'll discuss this homeostasis in the next lecture in detail and all the aspects related to that. But what is homeostasis? Homeostasis is the maintenance of constancy in the internal environment. We need to maintain a constancy in the internal environment. We are always in a dynamic phase, changing phase. And this dynamism, this changes, is taking place every now and then. And this dynamism, if you maintain the constancy, keeping this dynamism, then you are maintaining there is a propagation of life. If there is no dynamism, there is dynamism, but constancy is not maintained. We have to have the normal body temperature. We have to have the normal range of pH to function the different enzymes working at those pH. We have to have the normal body temperature. So this is all important. We have to have the normal osmotic pressure. Okay, so these are the things which are important, and these important things are needed to understand. We'll discuss homeostasis in detail in the next lecture tomorrow, so don't worry about that. And there is some historical perspective. History, you don't have to remember this. The classical era, the early modern period, and the late modern period. These are physiologists have divided this the era of physiology into these things. You don't have to remember it. The important things, some of the milestones in the history of physiology are these milestones. So Aristotle has started with relationship between structure and function. This is the most important basic information from where this turning point of thinking and understanding and science has started. A structure supports the function. Look at the structure of the cell, the structure of the skin cells, the structure of the liver cell, the structure of the nerve cell, the structure of the muscle cell. Allah subhanahu wa ta'ala has designed those structures so that they can support the function. And each cell has different entity, different structure, different contents into it, different compartments into it, different enzymes into it. Look at the cardiomyocytes. If there is a heart attack, the enzymes released from the cardiomyocytes comes into the blood. And that is what we talk about in the heart attack, in myocardial infarction. So this is how the structure, always remember, the structure always supports function. That's why we have different types of cell tissues present in the organ system. Am I very clear here? Similarly, the father of physiology was the person, Galen. He started to perform experiments to understand functions of the body, and that is why he is known as the father of physiology. Jean Fallon is the person, French physician, who termed from the Greek word, he coined the word physiology. And this physiology is the study of nature, and its origin and its progress. The first experimental physiologist was William Harvey. He was the first person who described the circulatory system and interaction of the body with the circulatory system, and he determined the blood pressure and all these things, circulation of the blood and how it goes on and so on and so forth. The father of modern physiology and experimental medicine is Claude Bernard, and he is the person who designed the term milieu interior. This milieu interior is the internal environment. We have 2 lectures related to that. My 3rd and 4th lectures are related to that, so don't worry about that. We'll discuss in detail. So milieu interior is the internal environment, and this is what related to the homeostasis. Constancy, maintenance of constancy in the internal environment. Okay? So we'll talk about that, don't worry. There are lots of branches of physiology. We have the cellular physiology and we have organ system physiology. We say GI physiology, renal physiology, okay, respiratory physiology, endocrine physiology. So there are lots of physiology. But what these organ systems are doing, all these organ systems are helping to maintain constancy in the internal environment. Respiratory system is working so that we do not accumulate more and more carbon dioxide in our body. Digestive system is working so that we are getting rid of, we only derive substances needed for our metabolism. We get rid of those substances which are not needed. Okay. Similarly, the excretory system is helping us to get rid of waste products. So on and so forth. Every system is helping us in maintenance of homeostasis. And that is how the whole systems are contributing, and the whole multicellular organism is maintaining constancy in the internal environment. So physiology can be, there are viruses, so viral physiology, bacterial physiology, how the bacteria surviving, functioning, and existing, and multiplicating and surviving these. If you want to study viral physiology is there. Cellular physiology, we have the plant physiology, human physiology, so on and so forth. There are more and more divisions are going there. And the other term, there is a true term, pathology and physiology. The pathophysiology is the junction of physiology and pathology. Physiology is the study of normal functions. Pathology is the study of abnormal. What abnormalities goes on into the different disease mechanism, under the different disease mechanism. So pathophysiology is the mechanism, the process, how the normal function becomes abnormal. If you start studying the pathophysiological mechanism, how the normal body develops a disease process. So this transformation from the normal to abnormal is the pathophysiology. So this mechanism, so pathophysiology is there. So we discussed lots of biophysical processes, how the things are moving and their changes affecting in disease mechanism, and that is the basis of clinical medicine. So the pathophysiology is the junction between physiology and pathology. So these are the two subjects, and this is the mechanism. Here we study all the biophysical processes which are taking place while we are moving from normal physiology to abnormal pathology. Let's look at some philosophical, approach to that. So how do we study physiology? Function versus process. So there are two approaches to the study of physiology. One is the function. So why? Why is the question? And the process, how is the person? So why? Say for example, if you take what we call as the theological approach, or we look at the purpose, why it is happening. If you address the question why, this is the theological approach to study physiology. And for example, why do red cells transport oxygen? Because every living cells need oxygen. So this is the answer to the why. But the next question is how, or the process. What is the process? How this oxygen is transported? So you need to understand that there is a presence of hemoglobin, and there is an interaction of reaction between the hemoglobin and oxygen, and then oxygen binds with the hemoglobin reversibly, and then it goes to the tissues, and then it delivers to the tissues, and then it binds with the carbon dioxide, and so on and so forth. So this is the mechanism. So we always look at theological how approach and why approach. So you need to understand, if you really want to know the basics of physiology, you need to understand why and how. These two processes are important. I'll give you this is a very simple diagram, and this simple diagram is showing the basis of glucose metabolism is pancreas. Pancreas secretes two hormones. I'm just focusing on two hormones, insulin and glucagon. One is responsible for decreasing blood glucose level. The other is responsible for increasing blood glucose level. If your blood glucose level, our blood glucose level remains rigidly maintains between 70 to 100 milliliters, milliliters. Okay? And this 70 to 100 milligrams per deciliter, okay, is important, and that is what it has to be maintained in this narrow range. Now the question is, if because of when we eat, if the blood glucose level increases above this level, so what we have to do? There is a hormone which secretes from the pancreas, from the beta cells. This is insulin. Insulin comes there. Insulin tries to push all these glucose into the cells, and that is how the blood glucose level decreases. If there is decrease in blood glucose level, so what happens? There is a glucagon comes, and it breaks the glycogen into glucose, and that is how the blood glucose level increases. So we have a normal balance between insulin and glucagon, and this normal balance is taken. And this normal balance is responsible for making the blood glucose level, maintaining blood glucose level within a normal, narrow ranges, rigidly. Okay, this is very important in order to optimally function all the body tissues. Otherwise, if there is hyperglycemia or hypoglycemia, then things will not work, and that is how the metabolism will be affected. Now the question is, if you just look at the mechanism here, if the blood glucose level increases after for example, when we eat, so what happens? This is one of the cells of beta cells which produces insulin, beta cells. Okay? So what happens? The beta cells has receptors, GLUT2 receptors. So glucose through these GLUT2 receptors, it goes into the beta cells. Once it goes into the beta cells, it is used for energy, and this energy produces lots of adenosine triphosphate, okay, through the TCA cycle. You remember TCA cycle? So what happens here? You will discuss TCA cycle in biochemistry, don't worry about that. So what happens? So the ratio of ATP or over ADP increases. Once the ratio of ATP or ADP increases, means more and more ATP is produced. What happens? There is a potassium ATPase, potassium channel, which is ATPase sensitive potassium channel is here. What this ATPase sensitive potassium channel is doing under normal condition, when there is less amount of ATP into the beta cells, it causes movement of potassium out of the cell. So what happens here? If it is, once the ATP level increases, increased ATP level blocks this potassium channel. Once the potassium channel is blocked, what happens? Potassium is not going out of the cell. So what happens? More positivity increases inside the beta cells. And this increased positivity under normal physiological conditions, inside the cell is negative, outside the cell is positive. So that's why we call it a polar state. What we call it? Polar state. But once the polarity means inside is negative, outside is positive. Once the polarity is lost, we call it depolarization. If the potassium is not going out, it has the increased ATP has blocked this potassium ATPase sensitive potassium channel. So potassium is not going out. If the potassium is not going out, inside the potassium channel remains inside. So this causes depolarization, and this depolarization changes causes voltage gated calcium channel. It opens the voltage gated calcium channel. Once the voltage gated calcium channel opens, this voltage gated calcium channel brings calcium inside the cell. Once the calcium comes inside the cell, what happens? This is the beta cells which is responsible for producing insulin. And insulin, once produced, it remains into the membrane bound structure. Once the calcium comes there, calcium binds with this membrane bound insulin vesicles, and then it releases insulin. This is the normal mechanism of insulin release. Am I very clear? So whenever the blood glucose level increases through the GLUT2 receptors, it comes here. Then through this more and more ATP is formed. It blocks the potassium ATP sensitive potassium channel. Once it is blocked, it causes depolarization of the cell. It results into opening of voltage gated calcium channels, which causes influx of calcium. Calcium comes in, calcium binds with the membrane bound insulin vesicles, and then it releases through exocytosis. Very clear. This is the simple diagram of how the blood glucose level is maintained. But this is not the case. And here, sulfonyl ureas and deanides also have the power. They also inhibit this ATPase sensitive potassium channels. It means once they inhibit this ATPase, I'll come to that. Look at this picture. When you do the GI system, when you will do the nervous system, when you will do the endocrine system, then you will have a bigger picture how this is glucose metabolism is, how the glucose is kept in a normal range from 70 to 100. Because not only glucagon and insulin are working, there are lots of hormones which are working in the body in order to maintain the glucose level in a normal limit. And this is the bigger picture. If you just look at these, we have the brain islet of Langerhans, which is the beta cell axis. We have the adipose muscle tissue axis. We have the gut, the male gut, gastrointestinal tract. We have the liver islets axis. There are lots of axes are working, and they are secreting different neuropeptides, different hormones, and those hormones have their effect either onto glucagon or onto insulin, and that is how they are maintaining the normal regulated control of blood glucose level. Am I very clear? Yeah. So once you go and discuss, so physiology is not a simple way. You have to look at it. holistic picture, the complete picture. And this complete picture is not the interplay of the glucagon and insulin. It is the interplay of these all hormones coming from different systems which are maintaining the normal blood glucose. If coming back to the same picture, what happens? We have already said that the glucose, if the blood glucose level increases through the GLUT2 receptors, it comes here, there is a metabolism, ATP is being formed, it binds with the nucleotide binding domain, and this is the potassium, ATPase sensitive potassium channel. And this ATPase sensitive potassium channel has, I have already written here, it has different domains. This is a receptor, it has different domains. So what happens? This domain is, if you just go and study, once the physiologists and biologists study that it is not only the ATP which inhibits this ATPase sensitive potassium channel, but there are also different other hormones which are inhibiting, different other medicines, other drugs which are inhibiting this ATPase sensitive potassium channel. And those are sulfonylureas and glanides. These are the drugs. So people who are suffering from diabetes, and what is diabetes mellitus? Increased sugar level. So what increased sugar level means? Because they either because of no production or low production of insulin. So what do you want to do? There is a type 1, no insulin is being produced. Type 2, less or moderate or less amount of insulin is produced as compared to the body size. Okay? So what happens? If we start giving sulfonylureas and glanides, this is the application side of how you need to understand how physiology works. And that is why in Nobel Prize, the award of medicine is physiology and medicine. It is because of this. The physiology of medicine, this is physiology of medicine. So this is how, so if a patient is given sulfonylureas and glanides, it will also inhibit this. Once it is inhibited, change in membrane, depolarization will be there. It will cause voltage gated sodium channels will open, will move this calcium into the beta cells, and then it will bind with the membrane bound insulin receptors, and will cause exocytosis of the insulin. This is another mechanism. So what you need to understand is, you need to understand the different contexts in which this physiology is working. So it is not only one part. You have to look at the bigger picture. While understanding the mechanism or the receptors and its domain, you need to, then the scientists, they came to know that, okay, it is not only the ATP, it is also the sulfonylureas and glanides. These are the substances which have the power to inhibit this, ATPase, potassium channel, ATPase sensitive potassium channel. So this is how they came to know, and this is how these drugs are being used in type 2 diabetes, where we can inhibit this receptor, and we can cause the release of insulin. Am I very clear? Insulin is also another definition of insulin is extracellular hyperglycemia and intracellular hypoglycemia. Under the effect of insulin pumps the glucose from blood into the cells, whether it is adipose tissues or the muscles or any tissue. But if there is no insulin, so this pumping effect or this pumping of glucose into the cell will be stopped. So what you have to do? So it results into hyperglycemia outside the cell and hypoglycemia inside the cell. So that is what another definition of, we can say, the extracellular hyperglycemia and intracellular hypoglycemia. Yes.
So the term hyperglycemia refers to low insulin?
No, the term hyperglycemia refers to the blood glucose level. That means increased blood glucose level. Hypoglycemia means decreased blood glucose level. So increased blood glucose level means if the person is, when we eat, if a normal person eats the blood glucose level after the digestion increases. So what under normal conditions, your pancreas will, beta cells will secrete more and more insulin. It will pump those extra amount of glucose into the cell. So that this glucose is available for the cells to extract energy. It is used as a substrate for energy. Make sense?
So high insulin, extracellular hyperglycemia. Or intracellular hyperglycemia.
Look at the term. If you say hyperglycemia, hyperglycemia means not high glucose level in the blood. Remember, hyperglycemia is high glucose level in the blood. Hypoglycemia means low glucose level in the blood. So we are talking about the blood. Okay? So don't relate it. And that's what I said. If there are two people, one is having normal, insulin production, the other is having no insulin production. If there is hyperglycemia here, the insulin will come up here, and the increased glucose level will be pumped into the cell. In this case, there is no insulin, so it will remain outside until unless you give insulin injection or you give these drugs. Make sense? So you have to understand. Yes, please.
So another point is remains the same as receptor.
I just want to give you another example. Have you heard the term ORS, oral rehydration salt? Have you heard this term? You never heard this? ORS, oral rehydration salt. Have you heard this term? No? Oral rehydration salt is the salt which is given in cases of diarrhea. And where you can, in diarrhea, what happens? There is dehydration, loss of water. If loss of water, dehydration, then what you have to do? You have to give oral rehydration salt. The oral rehydration salt has a combination of glucose and sodium. So why it is a combination of, and because of the use of oral rehydration salt, we have saved millions of children across the developing countries, because this oral rehydration salt is helping us to save children who are very susceptible for this change in electrolytic balance. Am I very clear? So ORS, you know what is ORS? Oral rehydration salt. Rehydration. Rehydration means opposite to dehydration. Dehydration, loss of water from the body. Rehydration, you are regaining water. But water is not regained unless you take it. So what this basis of this whole, the formation of ORS was made possible because of the discovery of these cells present into the lining of the intestine. These are the type of receptors. These are the type of receptors mechanism where sodium glucose co-transport or SYMPORT. Sodium and glucose are transported. If you give sodium and glucose, and sodium and glucose is present in every household, whether you have a small poor village of Africa, they have salt there and they have sugar with them. So if they mix salt and sugar, it is sodium and glucose. And they just put water and then dissolve it and give it to the children, it can save children. So this is because of the understanding of the sodium glucose SYMPORT or sodium glucose transport in the linings of the intestine. And what happens because of that? Sodium, if you give only glucose, this pump will not work. If you give only sodium, it will not work. You should give sodium and glucose so that it can work. Once it works, from the vital membrane, it transports this glucose, it transports sodium. Glucose goes through the GLUT2 receptors into the blood, and sodium, potassium, ATPase will pump the sodium into the blood, and that is how once the sodium goes there, it also takes water hydration along with that, and that is how the rehydration takes place. Yes.
Dr. I want to ask you why do you use a physical
Actually, yes, this is what, I mean, the pinch of salt and the fistful of sugar. The reason is, sugar, I mean, although you need 2 sodium and glucose, the pinch of salt, if you look at, what is the molecular weight of glucose? 20. What is the molecular weight of sodium? 22. If you take a pinch of salt, there are lots of sodium into it. So, yeah. And if you take one teaspoon of sugar, 180 molecular weight. So that is because of it. Make sense? So because of this, if you are those scientists who understood the mechanism of sodium glucose transport into the intestine, they know if we use this combination, then this combination, this is available in every house, I mean, home, in any place, in poor, I mean, people also have glucose and sugar, sorry, glucose or sugar or non-slime or salt. So this is table salt. So if you just add them with water, and then you can save a life. This is, so what is the understanding? Understanding is that this is physiology. This is medicine. And this is how you need to understand it. If you know the application side of this, if you have understood this, then you start applying it. Once you apply it, you can save millions of children. And this is the understanding, this is physiology. Themes in physiology. Structure and functions are closer, as I told you. Structure supports function. So whatever the structure of the cell, it has to, it is, Allah subhanahu wa ta'ala has designed it such that it will support the function, whatever the function is assigned. And there is a molecular interactions, lots of molecules are there, and their interaction is there. And there is a lot of compartmentation. The cell, usually, a eukaryotic cell has cell membrane. They have, I mean, membrane bound structures, membrane unbound, structures without membrane. So all membrane bound structures have their own environment within the membrane, and they have compartment. And this is what the understanding of compartmentation. Every eukaryotic cell has different compartments because of the presence of membrane bound structure. And each membrane bound structure is responsible for different, it has different functions. It performs different functions because it has an isolated environment within a cell. Am I very clear? So try to understand. Living organisms need synergy. Information flow coordinates body functions, and homeostasis we'll talk about it tomorrow. Level of organization. Atoms, subatomic structures, then molecules, then cells, then tissues, then organs. Organ system. And we'll talk about the multicellular organisms, and then we look at the species, and then we look at the interaction between the species and the cells, and that is how the physiology and ecology and cell biology and chemistry and biochemistry and molecular biology, all they exist together, and that is how we are learning physiology here. And this is how you can see that it is also extending from the molecular interactions up to the interactions between the living organisms. Organ systems of the body. As I've told you, we talk about the organ systems. So all the organ systems of the body are there to do one thing, to provide constancy in the internal environment. And this constancy in the internal environment is the guarantee to propagation of life, guarantee for sustenance of life. And we'll discuss in the tomorrow, how the different mechanisms and how it is working. So the best physiology is best learned. So if you understand the histology, macroscopic, microscopic, and the structure, then you can understand the function. Because its structure supports function. You need to understand what is the histological feature of these cells, what is the macroscopic structure of these cells, and how it is helping into the function. If you want to know the I should say the contraction of a muscle, you need to understand the histology of the muscle. and the arrangement of these bands into the muscles. And their, particular arrangement, if you do not understand, then how it will change in the contraction state, how it will change in the relaxed state, and that is the mechanism, the interplay, that is very important. So individualizing, memorizing individual relationship will never work in physiology. You need to understand its relation in the context of function, its relation, what are the consequences, what happens, what is there and what is not there. So you have to ask a lots of questions with your own self while reading articles, while reading things. So you try to understand the physiological process and try to understand the pathophysiological focus, because you are learning physiology. See, try to understand the basic difference. Teaching physiology to nurses is different. Teaching physiology to doctors is different. The nurses, they don't, take actions to, for the management of the patient. They just follow the policy. You say that just maintain the blood pressure, input-output chart, every 6 hours, every 10 hours, they will follow. They will check temperature, they will do it. The actual understanding is, responsibility is on the shoulders of the doctor. They need to, if they write some medications, if they are writing some interventions, medical or surgical, they know that what is the cause. They know the consequences. They know the pathophysiology. They know the etiology. They understand the whole bigger picture. So teaching physiology to nurses is different. Teaching physiology to doctors is different. In nurses, we do not explain in depth the consequences and relationship and its effect. But in doctors' case, you need to, because you need to know why this physiological mechanism has become the pathological mechanism. What is the pathophysiological mechanism? And why this disease is there? If this is the disease, what is the intervention? Medical intervention or surgical intervention? What are the consequences of that? If you are modifying, giving some drugs, what is the effect of the drug? What is the unwanted effect of the drug? So on and so forth. You need to have a bigger picture to try to understand. So this is the disruption of the normal state is pathophysiology. While reading, try to learn these questions. What is the likely mechanism? Likely cellular response, clinical processes, physiological readings, what are the effects, hemodynamic, pulmonary, what is the likely cellular mechanisms? All these things you have to understand and you have to learn while learning every part of physiology. And then once you have try to answer these questions, ask these questions, then you will learn what is physiology and you can easily understand physiology. Anything else? Any questions? Yes, go ahead. I'll post it. Right. I have a lecture right now, then I go. So guys, those who are put your attention on